I think we'll get started now. Welcome back to our first meeting of 2024. I realize that this is still early days of the year, so some people may still be on holiday if you're in Europe. So I appreciate you showing up today. As always, this meeting is live streamed and recorded in an edited form and made available. We're very pleased to have Jake Beal coming to talk to us today about data curation and modeling design. Given the interest of this community in trustworthy models, I think this is a topic of great importance to us. And so I hope people will have a lively discussion after the presentation. As always, Tom Helikar and myself are available to take comments and suggestions, Jim Sluka and Lorenzo as well. Please reach out to us as needed. We have a Slack channel, which just doesn't get used very much. There is a Twitter/X account, MSM Viral. Please use that hashtag. Help us advertise. There's the GLIMPRINT name and website. Our main communication source is still the IMAG MSM wiki page. There's an IMAG LinkedIn page as well, and of course our YouTube archive. Please help us bring this meeting to people's attention. We have the GLIMPRINT name and website. We need help doing things with it. Please help us work with it. We have that MSM Viral hashtag. Please help us with that as well. There are a few announcements. There is a staff scientist position in neural developmental dynamics available at NIH if people are interested. Certainly people might have postdocs or grad students who'd be interested in that problem. We are going to have our first steering committee meeting of the year tomorrow at 11 a.m. If you've been invited, you know who you are. If you think you should have been invited and you weren't to the steering committee, let us know and we'll add you to the invite list. Are there any other announcements from the room at the moment? I have a pre-announcement, which is that at Indiana University, there's going to be a set of searches in biomedical engineering and bioengineering, broadly defined. We're talking about potentially hiring 7 faculty at all levels of seniority in the next couple of years. I'm the search chair. The announcement won't go out now for another week or so, but digital twins and computational bioengineering will definitely be part of that. And so just be aware that there will be an announcement coming out for quite large-scale search in this area in the next week or two. Coming up, we have Adam Hollis speaking to us next week and 2 weeks. We have a talk on AI-based computational modeling for autoimmune disease development. We have somebody from the East Science Center talking about community model development and high-performance computing. Belinda Akpa is doing very large-scale molecule-to-organism prediction at Oak Ridge, which should be quite interesting. And so I think we have an exciting program coming up for the next few months. If you have people you would like to hear from, please let us know. If you've spoken before and you'd like to speak again, that's fine. If you'd like your graduate students or postdocs to speak, either for the first time or second time, that's fine too. We want to be a forum that helps develop careers and helps people interact with each other. So today we have Jake Beal from Raytheon BBN, and he'll be talking to us today about Agile data curation for modeling and design, which again is a topic we've heard quite a bit about over the years, and I know is of great interest to the community. I don't do fancy introductions, so I will turn over the screen share to you, and we look forward to hearing about this.
Thank you for inviting me today. As James said, what I want to talk about is about curation. I'm going to particularly talk about this with respect to applications in modeling and design. And the Agile data up front is talking about how we've been going about it and some new ways of trying to take things that have worked well in the software world and bring them to help us with some of the problems that we experience commonly in the scientific world around data quality, reproducibility, etc. So where I'm coming from for this is really out of the synthetic biology community. So you're going to hear a lot of that flavor in the sort of examples I'm talking about in my talk here today. And synthetic biology is, lets us do all sorts of really cool things, at least in theory, but it's really, really hard because we're dealing with biology and engineering it. And, so even though there are all sorts of wonderful things that we can do, much of the history of synthetic biology is littered with things that don't really work very well and things that work well in somebody's hands, but not in somebody else's hands, and a lot of dreams of how well we can engineer things that we're still trying to realize. So when I think about these challenges of engineering, a lot of it really comes down to combining information together that's coming from a lot of different diverse sources. So if we want to actually have effective biological engineering practices in that field, well, we've got to have good quantitative models of the systems that we're trying to engineer. And we're starting to get some of these coming out. But those models aren't going to do us any good if we don't also have good experimental data with calibrated measurement, putting reproducible units on it. Far too much biological data for far too long has been in relative or arbitrary units. And it's just not possible to mash up data and models across laboratories if everybody's on a different unit scale, because everybody's done their own relative units or arbitrary units. And we're starting to see methods coming out for that as well. I work with the NIST flow cytometry group, working with iGEM. We put together a whole bunch of stuff for calibrating plate readers. There's some great stuff that's come out for calibrating RNA-seq and other things. And also, it doesn't really matter if we've got good models and good measurements if we're trying to put together devices that don't work very well. So there's also a lot of work that's been going on over the years for giving us better tools for actually doing the engineering we want to do with. And there are lots and lots of different groups that have contributed nice tools to this space. But if you actually want to build a system out of those tools, you need to take a bunch of different tools and put them together, and take the models for the different tools and put those models together and make sure the units come together. So we've really got a big integration problem here of a whole bunch of different sources talking about things going on in a complex system. And this is why I think that some of the things I have to say here, even though I'm coming out of a somewhat different space, might turn out to be very relevant for folks in this multi-scale modeling community as well, because I think you have a lot of the same problems of a lot of different models and a lot of different data sources that need to be mashed up in an effective way where you don't rebuild everything. So biology is hard. Synthetic biology is hard. And a lot of times I hear people say this notion of sort of, well, maybe it's special. Is it possible for us to actually put all these things together? Maybe biology is just too hard and we can't do this. In general, we, as, scientists and engineers have learned a lot about how to manage really complex systems. I put a picture here of somebody, doing a pre-flight checklist in a military transport aircraft, which is an incredibly complex system. But doing this is, hard. It's expensive to do this, and maybe it's just too hard to do with biology. What I've found over the years, though, in all sorts of large problems, is that in many cases, biology isn't the problem. Biology is the easy excuse that we get to reach for when we run into what's often not a biology problem, but a biologist problem. With our record keeping, our data quality, etc. A nice example of this is the interlaboratory studies that we ran with iGEM. For those who don't know, iGEM, the International Genetically Engineered Machines Jamboree, is an annual event where undergraduate and high school students around the world, thousands of them, spend their summer doing a synthetic biology project and then get together in a big jamboree to be judged on the quality of what they've done and show off to each other and have a big party. It's a huge educational event, great fun to be involved with if you ever get a chance. And we started running some interlaboratory studies because one of the big things that iGEM tries to do is they collect, parts that folks have engineered and try to make it more possible for teams to build on each other's projects year after year. A lot of problems with that if you can't compare the units. So we started working on, plate reader and flow cytometer and trying to make protocols that not only could calibrate but could be done by high school students and undergraduates for a price point of, no more than $1 per kit. And 2016 was the third time that we ran it. It was when we started finally really getting things to work right. And we found something fascinating when we actually had good calibrants in the kit and some good standards for collecting things, that the wildly different units that we're getting over there on the left side, the red and magenta bars are showing the amount of variation in the measurements that people were getting of these very simple standard fluorescent constructs. And it's showing these geometric standard deviations on the order of 10. That means 4 orders of magnitude in the 95% uncertainty range of what a parameter value is. That's impossible to work with. If I don't know what a number in my model is to within a factor of 10,000, I'm toast. And we thought, okay, putting things on units should help. It didn't, not very much. It didn't help at all for the plate readers. Putting it on the units was also terrible. Putting it on the units helped a little bit for the flow cytometers. Normalizing, which is the other standard thing that people do. They say, okay, take your reference construct and normalize to it, and it'll all divide away. That was no good either. It helped a little bit for the flow cytometer. It helped a bit more for the plate readers, but we're still not at an engineerable level. But what we actually found was when we used the calibrants as process controls, not just to calculate the units, but to also say who screwed up using their machine. Who has their OD calibration, as they dilute it more, the OD goes up instead of down, or bounces around, or clearly this person didn't pipette well. Their positive control and their negative control look the same. Once we added in the process controls to do a little bit of quality control, bam, that's those green ones, the pruned ones. All of a sudden, we get things on the order of 2-fold standard deviation instead. And that's an engineerable value. Normalizing helps a little bit more, but not much. This is one nice example of where it wasn't the biology that was complicated. It wasn't the biology that was making it hard. It was that stuff goes wrong. And if you don't have something in place to catch it, it just blows the whole thing up. This is one nice example. We've worked with a lot of partner labs over the years. So BBN, we didn't have our own lab for quite a while. We got it one a few years ago. And so we spent a lot of time partnering with a lot of different people in wet labs. And every single time that we started working with a new wet lab partner, once we were applying our data quality requirements, after, we run into a problem where something had been lurking in that lab that had been going very wrong for a long time, and nobody knew it until the process controls went in place. And suddenly you had a graduate student or a postdoc getting very uncomfortable and very defensive, and the professor getting very upset. And, and after a while you work it out and everybody's much happier than before. But it's a bit rough because everybody wants to blame the biology. So what this has taught us is that we actually can do effective integration, but to do it well, we need good curation and process controls on the methods that we're using, on the metadata, on the units, on the statistical analyses, every single point of that tool chain is another place where something can go wrong, and an error that will cost 6 months or 5 years will be smuggled into a laboratory notebook somewhere. So I don't want to live that way. Now, there are a lot of tools that have been brought out there to try and help with this. Lots of people have been thinking about automation and about lots of different stages. In synthetic biology, we often think about engineering as coming around what they call the DBTL cycle: design, build, test, and learn. And a few years ago, me and one of my other colleagues at BBN we're looking at, like, okay, how good are the tools? What's the level of automation that we can reasonably integrate together in the lab? And we looked at each of these stages and the tools that have been published out there and that were publicly available, et cetera. Evaluated them against sort of an autonomy level stack similar to the one that they have for driving. And we found, it looks pretty good. There's great automation for design. There's great automation for laboratory equipment, for the build and the test stages. There's reasonable automation for the metadata and early data processing stages of learning and interpretation. But notice there's some little gaps in there. Those gaps are terrible. What comes out of the design isn't ready to go into a build. And in order to actually do these automation, they typically require a whole bunch of curation of the knowledge that's going to be necessary for this particular thing to operate. So we actually have a lot of pieces of automation out there and a lot of places where things get dropped on the floor and you lose all the benefit of the automation when you're going from stage to stage, or when you're paying the price for trying to start automation working on a new system in your lab. So, is this hopeless? We keep going, lots of good things, maybe it's hopeless. Lots of good things, maybe it's hopeless. Well, one of the things that gives me a lot of hope here is we actually have good representational tools available now for being able to talk about these whole tool chains and the integration across all of these different stages. In particular, one of the things, the communities that I've been involved with and, where James and I, meet in the standards meetings that happen. So SBAL is another member of the Combined Standards group, and it is synthetic biology open language, and it gives us some representational tools for talking about that whole design, build, test, learn cycle with respect to the engineering of biological systems. It's in many ways sort of complementary to modeling languages like SBML, which typically focus on the dynamics, and SBAL talks about, what do you need to know about the things that you would use to put those pieces together when you're thinking about putting genetic components together and putting those into context in a lab. I won't go into any of the details here, but just say that this is sort of our starting point for thinking about when we're in these engineering spaces, how are we going to knit the pieces together? And a few years ago, starting in 2017, there was a big DARPA program called SD2, which pushed a lot of this stuff forward. SD2 was, I think, systematic discovery and design. And it was basically, folks at DARPA said, let's take laboratory automation and, big data and AI and put them together in the cloud to make science faster. And that then immediately ran straight into these integration challenges where, you have the folks with the designs over here, the experiment descriptions being tossed around in, Google Docs. That was an improvement over Slack, which was where it was first happening. Talk about a self-erasing, a protocol notebook. And then, of course, somebody actually runs the stuff and the data and metadata come out, and all these pieces need to be put together. You can't analyze your data without being able to integrate it with the metadata about the designs and the experiment plans that would put in. So over the course of this 4-year program, we built a whole bunch of complicated bits and pieces that hooked all these bits together, using SBAL as the interlingua and the SynBioHub system that Chris Myers at University of Colorado and his team support as the knowledge store for that most of these things talk to. And I'm not going to tell you anything about this, except that it worked and it was way too complicated and it was not very good because we were building these tools on a basic research budget. So at the end of this, we learned a lot of lessons about how to do this. But one of the lessons I also learned was that we shouldn't build this software. We need this software, but we can't build it ourselves because there's too many pieces and the budgets are too small. So I followed the advice of from one of my favorite books, The Mythical Man Month, about software engineering complexity, which is, build one system to throw away. Because what you learn from it will allow you to build something else much more elegant later. And so we threw away everything that we built in SD2. And because we realized that what we were doing was slowly trying to reinvent a lot of the things that already lived in the Agile software tooling world. And in the Agile software tooling world, there are billion-dollar companies which are backing these and pouring a huge amount of money into making them really, really solid and really, really good. And so if rather than building software ourselves, we can figure out how to wrap these tools and put little adapters on them to let them talk to biology, then we have a much better chance of getting effective and functional curation out of here. So all these wonderful things of, version control, build automation, checking for errors with continuous integration, all the, social and community interaction portions, all of that is things we're sort of creeping our way towards. But since there are a lot of nice biological knowledge resources out there, a lot of those can be made version control friendly and have, look like text. So what we found in particular is that using SBAL again as a rendezvous point, this let us take the things we were talking about in biology and let them be something that these distributed version control and of the rest of the Agile software systems could be treating like code and like log files. So an experiment description or a biological system description, a genetic description, that basically plays the same role as code. Things that, when you have experiment traces, data, that's something that plays the same sort of role as log files. And if we build a little bit of adapter code, then we can have the systems help us maintain our integration and automate all the checklists that we need in order to operate more safely and focus on the science rather than focus on cleaning up messes. So what I want to do with the rest of the talk is tell you about 3 cases where we actually did that and it made a big difference for projects that we were doing. The first case is a straight up modeling example. We were partnered with some folks on an NIH project on safety switches for CRISPR-based gene editing. As you may know, CRISPR-based editing is being applied for a lot of potential gene therapy targets, but you have to be careful with it because if your CRISPR system stays around too long, then you can start getting off-target editing or start to build immune response. So you really want this system to get garbage collected after a certain amount of time. However, if you shut it off too soon, then you don't get the therapy. So you need a tunable approach to self-deletion. There's a very elegant way to have the system self-delete. I've got a diagram down here. You've got your Cas9 for running your CRISPR system, and you've got, in purple, here's the therapeutic gRNA that's going to actually do the editing on your target. And then you add a second one that targets the construct itself, so that one then just self-shreds. People had already proved this out, that this works, but it's really fast. I mean, it basically shreds itself as fast as it exists. So what we were working on was trying to figure out, how do you add delay on when your self-deletion gRNA turns on in order to make a tunable delay before self-deletion. And we were looking at several different ways you could do it through transcriptional regulation. So say maybe put a chain of activators or a chain of repressors in front of it, or through recombination. You can have you use something like the CRE-lock system to flip the promoter, and then it'll only turn on once the promoter has actually been flipped into the right position. So we're working with these folks. There was a grad student doing a bunch of modeling in MATLAB, and we spent about 4 months slowly iterating, trying to figure out how much the designs that we wanted to investigate, how did they relate to the equations that the grad student was writing, and how did those two relate to the MATLAB code? And it was just, we had about 4 systems that got investigated over that time, and we didn't actually believe the results from any of them. Because we kept finding, here's a place where the MATLAB code doesn't actually follow this equation, and this equation doesn't actually look quite right, compared to the system diagram. So we gave up, and we said, okay, we're not going to write any equations. We're not going to write any MATLAB. We're just going to generate it all. We're going to take those whiteboard diagrams, encode them in SBAL, and use the SBAL as the starting point for generating. And then if we have, we can recombine in different ways and automatically generate the right things. Now, we had a lot of bugs in the code, but they're systematic bugs. And when you wipe out a systematic bug, it doesn't come back again, at least not the same way. So we took a month or so to build this pipeline. And what we ended up, we had basically simple Python scripts where you would describe what's the set of systems that you want to investigate. We were using GitLab for this. So you check it into GitLab, and then GitLab has its own built-in continuous integration pipeline system that we set up scripts in that would run that Python to generate SBAL files. And then those SBAL files would in turn generate a LaTeX with all the equations in it that would eventually go into our supplementary, but would also allow us to inspect and see, do we believe these equations or not? And it generated MATLAB code, which, because we couldn't run MATLAB on the server, we ran with the Octave free alternative instead to automatically regenerate the plots. And then we also took advantage of GitLab's code review system to review the model iterations. So, my junior colleague who was doing a lot of the actual coding on this would, we would meet with the biologist who has no idea about the coding, has a lot of idea about CRISPR and mammalian cells. And we would talk on the whiteboard. My junior colleague would go off, make some tweaks on the models, send them into a pull request. Once it's validated that the model is at least, working enough that it's passing the tests. I would review it. We decide, yes, we're okay with what this is doing. And we've got a whole batch of graphs to take back the next week to talk with the biologist again and see, are we happy with where this is going? So we basically got rid of the whole part of trying to figure out the relationship between the whiteboard, the equations, and the simulation code. made that part very rapid and trustworthy, and let us start actually exploring this space much faster. What we ended up doing here was, coming from the, I think I said 4 before, 5 initial architectures, we were able to start doing a combinatorially driven exploration of all of the possibilities that would fit within the size of cassette being considered. Ended up coming out to 31 different options. And we could generate all of these and be sure that we were covering everything. And we were able to explore these to a much greater depth as well, with all sorts of different combinations of parameter values. And one of the things that was a great surprise here was the actual circuit architecture that we found was effective, this activator to Cree-on architecture that you see here. This is showing different tunings of it that, in our models at least, would give us anywhere from a day and a half to a month for clearing 50% of the system. That wasn't on anybody's radar when we started. We didn't think this was a system worth even thinking about. But the combinatorial exploration led us to it over 2 months of about 2 iterations per week of model adjustments. And at this point, this is back off in the biologist's hands. I don't know if they've managed to actually test it, since that particular NIH project was coming to an end, and funding, etc. But we were able to put this out in a well-received publication that had a truly massive supplementary information with all the equations for all 31 of those systems, and I pity anyone who decided to actually try to read it all. So this let us go much faster and much more in depth than we were able to before that. Another example was working with iGEM. So again, spoke a little about iGEM before. Here's a picture of the about 350 teams that were in 2021 scattered all over the world. One of the big things that iGEM does is, as it's been collecting these standardized DNA parts on plasmids, it builds a distribution that's shipped to every team that participates. That's one of the big things that your entrance fee gets you, is hundreds and hundreds and hundreds of parts that are likely to be really useful for you. And they've been accumulating for a long time and had all sorts of issues with their old collection. So in 2020, they took advantage of the fact that they had to shut down their lab for a while to literally incinerate the old distribution and start rebuilding it from scratch. And came to me and the other folks on the volunteer engineering committee for help figuring out what should they put together to send to the students. So this is a committee with a lot of people in a lot of different places in a lot of time zones, a lot of very different sets of expertise, and somehow needing to collectively design hundreds of kilobases of plasmids. So we put some Agile data curation together to try and simplify this some. So in this case, we used GitHub and GitHub Actions rather than GitLab. And our sort of coordination point was spreadsheets. We made a template that could be translated into an SBAL description of a collection and a build plan that in these templates, you basically could say, here are the basic parts that I want to get. Here's how I want to put them into plasmids or into combinations. And it knew about a whole bunch of different data sources, like NCBI and SynBioHub and the iGEM repository, so that you could just give it an accession and it would go out and retrieve the information for you as part of the GitHub action. So people then were not playing with sequence editors at all, and all the problems that often happen there when you're passing things back around. They were talking about here's my inventory list that I want to have. And oh, I found some of these things over here, and okay, this didn't have some accessions, so I uploaded a couple of FASTAs, and we just refer to them and so on. So we were able to then, importantly, first validate that what was in there made sense, because people would typo things, people would put in a different accession than they meant to, people would use something in one place and then not in another, or they'd put duplicates in. So we started by just making sure that the description was coherent. Doesn't matter whether it was scientifically valid or not, but if you're saying I want this part here, you'd better have a copy of that part somewhere so that you can refer to it. Simple, stupid things. And also, every time that it went through this validation, it would also produce an SBAL description of the collections, and then put them all together, generate a simple markdown description for each collection, combine it all, and then export it into GenBank for describing all the plasmids, and also into FASTA build plans describing only the inserts that we were asking the synthesis company, Twist, to put into each of the plasmids. So every time that somebody made a commit and it passed validation, we also had an update of a ready-to-go order of this is exactly what we want to send in as the synthesis order in order to build all of this stuff. And again, using pull requests and change reviews, we also were able to have people have a discussion about what they thought about the proposals that other people were having to change. Somebody says, I want to use this fluorescent protein instead of that one. That goes on a pull request. We record, we have that discussion. And okay, you can't diff the Excel files, but one of the other things the pipeline did was it exported the Excel to a CSV that does diff nicely. So people would review the CSV and they knew that, and you could look at exactly the diff. This is exactly what, my colleague over here has proposed we change. So we stopped focusing on the sequences and we started focusing on the content. What's the set of things we want to give people? And how do we know that these are good choices to make? A little bit more about, there was a whole set of stages here. I'm not going to go into this much, just we had a bunch of micro utilities that went from sort of very high level whiteboardy designs all the way down to, FASTA by stitching pieces together and expanding combinatorial descriptions and so on. So when this actually went in February of 2022, there was about 0.5 megabase of synthesis, and with hundreds of parts involved, and the bulk of the development of the system happened in that month and a half before that. We were working on the tooling in the fall. And so there are some pieces we put together there as we were working that out. But almost all of it was happening at this very fast pace. You can see that's about 2 pull requests per day, getting reviewed and merged, etc. This had more than a dozen contributors across a whole bunch of different institutions in different countries. And one of the things that made me very happy was that, again, saying, use somebody else's user interface. GitHub has a pretty good user interface. And so most of these folks were not programmers. There were a few like me who were, but most of these bench biologists had never even used Git before. And we were able to get them up and running in a nice, professionally maintained user interface and skip around a lot of that. Last example I'll go through quickly. So we've seen a design one with the modeling. We've seen a build one with the iGEM parts curation. Now here's a learn-focused one for analyzing flow cytometry data. This is one of the common pieces of pain we have. There's some folks at University of Tennessee that we're working on plant engineering with, that we've done this work with. And basically, we're using the same sort of technology that we did with iGEM, but now rather than having it integrate sets of genetic design files, it's providing the metadata for analyzing a set of flow cytometry files. And the workflow there basically begins with the same spreadsheets that we're using with iGEM, talking about what are the parts and how do they get stitched together into constructs. But then we add one more sheet that says, how did those constructs get combined with reagents in sample designs? And then what are the flow cytometry files that got associated with each of those? Which then, this is still a work in progress, but our preliminary tests are showing that we can now get this to be a more smooth process. My experiences with flow cytometry is analysis is easy. Setting up the analysis is what takes weeks. So there's some examples of this working. And it's working for synthetic biology. Was it really about synthetic biology? In fact, was it even really about biology? The last point I want to make here is that this should be applicable to a lot of fields. The really key requirements are that we have some good modular boundaries in our knowledge base. This is something that is actually very important with SBAL. It lets us do semi-closed reasoning. If you're using sort of a totally open world, then it's very hard to actually do much reasoning with it, because you never know if there's a server with a document somewhere that provides one more assertion that's going to change everything. Having composition and traceability in order to be able to track these relationships is really important. We use a tool called SBAL Factory to make it easy to build extensions on top of the libraries that we've got. And there's some nice tools for syntax level verification out there. One other thing that's really important is making sure that your knowledge representation is version control friendly. This is something that RDF in its default serializations isn't. XML also not so version control friendly in many cases. But if you have RDF, you can turn it into the N triples format. And if you sort that, then it's beautiful for version control. It's the worst, dumbest format that you can possibly imagine, but it works really well. And you also do need to be able to ground your terms well. And for that, for us, ontologies are good. And I think for the biological modeling community, I think you're probably pretty deep in ontologies as well. So if you just have a set of these ingredients, you should be able to apply these same sort of ideas into all sorts of other areas as well, not just the examples that I had. So in summary, the experience that we've had is that the really key integration gap in lots of biology-related workflows is maintaining your curation and appropriate configuration information. Using these agile data curation approaches really helps us bridge the gap, because we don't have to build our own tools. We just have to figure out how to leverage the things that billion-dollar companies have been pouring lots of funding into making really good. And we've been able to do this in a number of projects and think that it ought to be usable for a lot of others as well. And with that, I will stop sharing and we can move over into discussion.
Okay. Questions and comments? I know you touched on a lot of issues that are of interest to some of the people in the room.
You went from a very complicated system, as you said, didn't work very well, to what to a neophyte user looks like a very complicated system that worked better.
I would say our system is not complicated, and that's why it works better. There's a very complicated system, but that very complicated system is maintained by thousands of programmers paid by folks like Microsoft and Amazon. And And yeah, you get lots of, weird things that break, but they're weird things that break for the entire industry, and people come in on their weekends to fix them.
Sure, but first of all, going through GitHub is a barrier to anybody that's not a programmer. And then the idea that you're starting on Excel worksheets, and you're pulling SBAL definitions, and you're using ontologies to precisely define bits and pieces. And you're going up and down through a work stack with different tools being incorporated. To what extent is that actually doable as a routine matter?
Mm-hmm.
In not only systems biology, but part of your whole point was, this is a problem in all of biology, period. Not just systems biology, not just engineering systems and things like that, but can people measure proliferation rates in tumor cell lines in culture and report them accurately?
So you've got to have somebody who really knows the tools in order to set up your automation. That you absolutely do. But one of the things that I thought was really important to me about the iGEM project was that you talked about using GitHub as a real barrier to biologists. It's not. The fear of using GitHub is a real barrier to biologists.
No, I disagree with that. The barrier to a biologist is not that GitHub is difficult. It's that it will take you a half a day to get, even remotely capable. And then you won't use it again for 9 months. And it will take you another half a day to get remotely usable in the system. And then you won't use it again for 9 months. So you say a system is easy to use only if you use it constantly. And when you use it and don't use it and use it and don't use it, you spend all your time relearning things you've already. I must have learned R at least 9 times in the last 10 years.
So let me give you another example of people. So yes, if you never use a tool, you may forget how to use that tool. It's not like riding a bicycle, but the learning curve is not too bad. One of the things that iGEM did this past year in 2023, for a long time, so all the teams build, permanent web presences that get archived after. And they had it being run on MediaWiki forever, and that was not scalable for many different reasons. This year, they switched everybody over to Markdown and Git-based backend that did auto-publishing. And so they taught all of these high school and undergraduate bio students how to use Git. Now, some of the teams had only some of the students learn, and some of them, stayed away. But anybody who can actually learn how to pipette well can also learn how to use GitHub. The question is just having a workflow that you want to use. And if you're doing Agile data curation, then you will be using that workflow on at least a weekly basis, if not more. Look at the paces that we tended to be working at, because we were having conversations about the science assisted by the tools.
I think that's a critical point that you're making, which is Jim's point is that if there's a barrier, people may not use it enough. But your point is that it's useful. And so we always would say it gives immediate practical value to people. So there is a payoff, a direct payoff from learning it. It's not just a cost.
And, the first time that somebody catches an error that would have cost them 3 months in a lab, they never go back. I see Kyle's got a hand up.
Great. Kyle, please.
Yeah, the issue of it's hard because I don't know how. And I don't know how because I haven't tried, because I'm afraid I might not like what it does to what I've already done. And I listen to this enthusiastically because if you're going to be in the modeling business, and if you have any concern whatsoever about the credibility of your work and the time and energy you spend doing your work, and you don't do this kind of tracing of the data, the sources, and what happens, the evidence, coming up with the evidence of credibility, when the FDA is looking at this stuff, you build a model, you want to make money with your model, you ain't going to make any money with a model unless you can produce the kind of documentation that was just described leading up to how the model was done. And if you don't like doing that, then you're not going to be able to be a modeler anymore, because nobody's going to give a damn about the trash that you turn out when you can't explain how you critically managed and treated your data and your inputs. I love that. Thank you.
Thank you. Coming into this as a programmer, I have a very deep relationship with regression testing. I don't trust anything that I do because it's all complicated. Even a simple thing is complicated. And, I sometimes with the folks that I manage on projects, I give them, there's two maxims I have. One maxim is that if it hasn't been pushed to the shared repository, it doesn't exist. And the second maxim is that if it's not being validated with, continuous integration regression testing, then it's broken. And I think, we need to accept that that's true of anything complicated we're doing. Now, Kyle, I think you had your hand up before.
Yeah, well, I think congratulations on not giving up. And I think you really advanced the practice by using source code control methods and stuff like that to basically create something you can verify, something that you know how you made your steps along the way. So beyond resistance to adopting tools, which we already kind of covered, it's a big deal. And even trying to start continuous integration on an existing project is super hard. It's like at the beginning, it's easy to set up something and build that in. What were if you were going to do this over, say somebody convinced you to try this again in a greenfield, how would you go about it, and what would be the things you would do differently if you had that opportunity?
I think the really key thing that I've seen is there's a culture you have to build. And this, I've run down biologists a little bit, but actually, this is a problem in software engineering too. Most software engineering does not take good advantage of these tools. There's a culture that you have to build about how people work with things. There is a tendency for a programmer to basically say, this part of the project is mine. I'm going to go off into a hole for the next 3 months, and I will come back with something beautiful. This is very closely analogous to the graduate student who has been given a project and says, I'm going to go off into the lab for 3 months, and I'm going to come back, and I'm going to have a graph that's beautiful. And it goes very poorly for the programmer, because there are so many ways that having more people put eyes on what you're doing earlier on can make it better and let you go faster, even though it feels like you're not. There's another project that I'm running right now, I didn't talk about, but it's a DNA screening project. We have actually a commercial tool that's out there deployed in the real world. If you order DNA from IDT or various of our other customers, our software is what checks to make sure that you didn't order smallpox. And so that's something that we have to have right. And it has to be maintained right in the face of consistently changing landscape. And one of the things that's been an important cultural shift in that team over the time we've been working on this product is building a shared understanding that looking at somebody else's code is real work. Doing a code review is actually, we've recently even been talking about the first priority you should have in every day is not doing your own work, but reviewing other people's pull requests, because that's actually incredibly valuable. It also builds awareness across the team. It's a way of knowledge sharing. And so it's building a culture in which you're taking small steps, you're sharing knowledge about the steps, you're testing your small steps, and you take as a first priority in knowledge creation the review of other people's knowledge creation work, that's real work. That's not just a cost. That's real work, and it's really valuable to both you and them. That's a cultural shift that I would like to see start getting out there through the life sciences, because that dramatically transforms how an organization operates.
Yeah, I call that like prophylactic shame, where you're like embarrassingly sharing your work product, and then you get used to it, and then you don't feel as threatened or embarrassed.
Mm-hmm.
And if you can get people in the habit of freely sharing things, especially when they're breaking the code and stuff and the processes, it really can kind of train the organization or the culture in kind of controlling damage versus damage control. You know what I mean?
That's a great way of putting it. Yeah. Yeah, when we spend time repairing, this is the sort of thing where I say, there's a lot of things that you can save a couple of hours of modeling with a mere 6 months in the lab.
Yes, I remember a friend who used to say, many months writing computer simulations can save several hours in the library discovering what somebody already did.
Mm-hmm.
So yes, that's a hard lesson, but one that humans seem to be poor at learning.
Well, that sort of shared awareness is very difficult. This is another piece of the curation puzzle, I think. How do you make information available where it's needed? How do you surface things? And a lot of that can't be done in an automated fashion. And this is, I'm, sort of speaking out of school as a computer scientist with some AI background, saying, no, my discipline is can't help us enough. But a huge amount, because it's ultimately in here that matters for the decision making. A lot of it really does end up being networks of expertise and information sharing. And this is why I really like these agile approaches, because they actually, you put this whole bunch of stuff into the infrastructure, and what it lets you do is submerge the technical issues and let the interactions between the human experts across disciplines be what's front and center.
One of the problems with Agile is that it works very good in small teams and within little narrow areas. And it also kind of gets things done and avoids having to build a master architecture first in a lot of ways. But if you've got lots of teams, and in your case, it seems like they're all doing the similar, almost the same thing. But if you got a lot of different teams, they have to extend it with something called SAFE or some other thing to have a holistic view of the whole thing. And it may not apply to teams of small labs, but it applies in a bunch of organizations that maybe are doing different things in a coordinated way.
And this is where it becomes a matter of standards.
Yeah.
So this is something that's been going on a lot with the NIST flow cytometry working group. That's an enormous working group. It's got something like 100 different organizations in it, across instrument makers, reagent suppliers, regulatory agencies, lots of different pharmaceutical and therapeutic companies, and everybody's figured out that sort of the key thing that's going to allow them all to communicate better is to be able to have standards about how they put units on data and how they believe what somebody's telling them. And I think there's so many different types of standards. We can get very hung up on sort of deep, heavy standards, like a format like SBML or SBAL. is a very costly standard to manage. But a lot of the actual interfaces can be much, much more lightweight, and they just, there's a lot of reasonable options that you could choose. And how do you rendezvous with one? The thing is you have to make it work is you have to have people who are actually motivated to communicate. But more than that, I think the biggest thing, and this is one of the other big lessons I've learned, is that you never get adoption unless the benefits are local too. One of the problems I think, we've seen a lot, with the SBAL community, I think the modeling community sees it also, is that if you share a model, other people benefit from it much more than you do. Now, the nice thing about something like Agile data curation is that you benefit from it right now immediately, but it also means that you're in a position to just hit publish at any point and have other people benefit too. So you flip that value equation and say, the quality control that I want is not for other people. The quality control I want is so that I can move fast. And oh, by the way, because I'm using standards, it's going to help other people too.
I'm sorry I have to go, but I just wanted to say thanks. I really enjoyed your presentation.
Okay. I think, I mean, I find what you say very inspirational. I also you started with this, that nice diagram of all the processes that need to be described and sort of the despair of trying to do all of that on your own. And I was thinking of the EDITH standard that's being developed now by the VPH community for digital twins, where they're basically describing all of those components that were in that beautiful process diagram that you started with. In our world, we're trying now to try to build a more of an open source community around multi-scale modeling. And it's already wasn't easy to get where you are with SBAL and SBML and the other data standards, for example, for genome sequences and so on, that exist already for omics and synthetic biology. I was sort of trying to mentally make an inventory of all the missing pieces. You had that nice list. You need to have some ontology basis. You need composability. We're pretty far from having any of those things, and it's hard just the way in your cycle. None of the pieces really. They all, you have to have the pieces have individual value, but they have the greatest value when you have the whole cycle assisted. And so I think one question for me is where do we start to try to bring in some of these practices, because we're not going to be able to do all of it at once because we don't have all of the enabling components yet.
So I guess the key thing that I see, one of the real challenges with an open source community, we were all, there's a lot of wonderful dreams that we've had about how open source can let us do fabulous things for free, on the backs of people in garages and basements. And an open source community only actually thrives when you have people who are financially incentivized to put things into the open source community. In biotech right now, we do have a real problem, especially in the laboratory information management space of everybody wants to be Facebook. And so they're going to try and lock down the pieces because they haven't figured out whether they can make money by taking a more Amazon-like model of, owning pieces of infrastructure and getting money through scale rather than getting money through, capture and rent extraction. So I think that, so it's going to be hard to bootstrap. I think that what we need to find, again, I come back to this notion of you've got to be the one benefiting. One of the reasons I do a lot of work with iGEM is because it's a community that has critical mass and a lot of motivation for doing these types of things. So, on the iGEM engineering committee, there's, several dozen people who are all putting in little tiny amounts of effort, but have a lot of motivation because something or other has caused them, whether it's education or, that they're It's, aligned with what they're doing in their PhD or what, but there's sort of a critical mass there. And if we can get a loop going there, it'll be powerful enough that then it can spread in other places. So I would say in modeling also, look for where is there something that's large enough, that's small enough that you can do all the pieces, but large enough so that it's going to have critical mass to sustain and start moving outwards. Because you don't need to have everything. You just, again, sort of the Agile thing, what's the minimum viable product? Getting something that's going all the way around the loop. And then you can start saying, oh, well, maybe we can do this thing too. Maybe we can do that thing too. Maybe we can clone it over here. And then the key thing is just to make sure that you end up expanding rather than forking, because that's the other thing that kills open efforts.
Yes, Jim had a nice cartoon on his door for many years of, we have 12 incompatible standards. We need a standard that will encompass all of them. And then the next slide is we have now 13 incompatible standards.
Yeah, standards building comes later. First, you have the community. And if you have people who are standards minded, then the things that they build can be things that are sort of standardization friendly. So, use the ontologies when they're available. Don't rebuild a new one. Whenever possible, use something somebody else has built that has a life outside of your group.
I think this is a social thing, though, because literally people in our community would still rather write their own hard-coded solvers, for example, rather than use someone else's solvers. Now, some of this is because there isn't the quality of testing and assurance for a lot of the software components that you would expect in industrial level software. But some of it also perhaps reflects that graduate student mindset, which is you want to have control of everything. But we know, from the experimental point of view, despite all of the variability and complaint and things you were talking about the difficulties of doing experiments, and I know the VPH people tried to do experiments measuring biomechanical properties of tissue, and the values of, say, the Young's modulus of cartilage came off were varying by factors of 20 depending on who was measuring it, which ought to be a relatively well-defined thing to measure. So it's not, again, on the experimental side, this happens too, but we're almost at the level of every time you're going to do omics, you build your own reagents. In other words, it's not only that you have your own protocols in the lab, but you don't order from Sigma. You actually create your reagents from scratch. And I'm not sure, of course, if you're developing reagents, that's fine, if that's your technology. But I don't know how we move from the idea that everything has to be under your own control to the idea that you focus on the things that are the value added, which are the models, unless, of course, your specialty is developing technologies for certain purposes. But even those don't get shared. They don't have standardized interfaces. They're not reusable. And so if somebody invents a new technology for a solver, you can't use it. You have to then read the code and rewrite it from scratch based on the algorithm descriptions. And so I don't know how mature our community is. And maybe we can talk offline at some point another time about getting some of your insights. I know you at Combine, you're familiar. You've talked to people like TJ and others. But I wonder if we can somehow begin to develop some of the sophistication. As you say, what you're doing isn't rocket science, it's using basic technologies. But from our point of view, that's still pretty far away, I think.
Yeah, I think it really is, it needs a group to seed it. And I think the really important thing is figuring out the selfish incentives of how is this going to let me be a better modeler who eats other modelers' lunch and gets tenure faster than they do. The drive to do everything yourself, I think, is something that academia pushes to some degree. As a graduate student, your PhD needs to be unique. Graduate students tend to get pushed away from one another. And then it happens again, as a postdoc, you need to differentiate yourself in order to get a faculty offer. And as a faculty member, in order to get tenure, you must be unlike all other faculty members. So there's this sort of, magnetic repulsion that happens to people, which I think is one of the things that culturally helps to try and push this. But I think, I really want to appeal to people's greed. If I use tools, then I can move at 3 times the speed of my competition and they won't be able to catch up. I'll publish faster. I'll get more grants because I have the power of automation. But you got to invest to get there.
I think it's a great point. The bootstrapping is hard. Once you have good tools and you can actually deliver on that promise of helping, I think then it does become self-sustaining.
It'd be interesting to have an offline conversation about like where might be some good places to seed it.
Are there other last questions for our speaker for today? We've had a good discussion so far. Lorenzo?
Well, mine was just a comment. I come from the in vitro world mostly, and I have a passion for modeling. And I always design experiment to assess certain function, etc. And I cannot do anything but endorse these principles and ideas of agile planning and standardization. You mentioned flow cytometry is a word that has been standardized widely because it's used in diagnostics. So it had a little bit push of industry, but there are other domains where this can be done. And also using tools like Git for certain aspects, but also there are other formal repositories that can be used for standardized biological experiments. So where everything is reported, and when this pass through the community, I believe that it will also be easier to integrate in vitro, in vivo, and in silico experiments so that they can deliver the best product possible for everybody, that is scientific discovery or clinical impact. So it's just a comment more than a question, but I think I very much agree with all that you said.
That you appreciated it.
Any other last questions or comments for our speaker for today? Jim, John, anything to add? Okay, well, we had a pretty good conversation. I appreciate that. Thank you very much for being willing to come to talk to us. I hope that this will inspire some people to go out and think more seriously. I think there are a lot of people here already thinking fairly seriously, but maybe inspired to think more seriously, the wrong word, inspired to try to put in practice more some of these principles that you've enunciated. And with that, I think I'll call the meeting to a close for today. Thank you again for coming.